Okay, I got my cue here. Good afternoon, everybody. I'm Johannes Früher, founder and president at Lab Central, and I welcome you to our fourth webinar, Innovation in Biotech in the Times of COVID. Today, with an exciting panel, we are going to be discussing the questions of testing around coronavirus. And I welcome our panelists for this afternoon, Professor Michael Mina from the Harvard School of Public Health. Michael is also a pathologist at the Brigham and Women's in Boston, and his expertise is mostly in epidemiological surveillance and high throughput technologies to combat and to trace outbreaks of disease. And of course, he's at the front lines of this now. We will be interested to hear how your life has changed, Michael, since the coronavirus hit. Also with us this afternoon, Professor Galit Alter from the Ragon Institute. Galit is an infectiologist, and typically her research is around infectious diseases such as HIV and malaria and tuberculosis. And Galit's life has also changed quite a bit since she was tasked to put up a large-scale testing effort at the Broad. And last but not least is Irene Bosch, a biologist originally out of Venezuela, and now an entrepreneur and founder of one of our startups at Lab Central, E25 Bio, who we had actually on a show before. And there, Irene was explaining to us how her company was developing a rapid test for Zika and dengue. And E25 Bio has recently pivoted to add a testing module for coronavirus as well. So we look forward to hearing about that. Thank you all for joining us. Maybe let me kick it off by asking you, Michael, about, we hear so much about testing in the news and on all channels. What is testing, and why do we even care about testing in the face of a disease like this?
Sure. So testing is obviously, I think you're absolutely right. Everyone has heard about it. A lot of people had never heard the word PCR or nasopharyngeal swab and all of these words that are now common lexicon for Americans and people across the whole globe. Testing is, we usually interact with testing in a clinical sense. And that's if you go to the hospital, you have to get tested for infections. If you go to the doctor, you get tested for sugar levels, whatever the test might be. In this case, testing extends not just to the person who is being tested, but actually testing is also an integral part of how we deal from a public health perspective with both monitoring individual health, but also monitoring population health. And so testing in the way that we've been thinking about it has kind of happened in this epidemic in really two phases. I think of, if we take a very United States-centric view for a moment, just to constrain the discussion, if we look at February, I would say that February and March were sort of the months of PCR testing, and these were the months when the whole world and in the United States was getting on board with doing molecular PCR testing, and this was really a test that is for personal health. And I shouldn't just say PCR testing in particular, since we have Irene on the line. But really, this is viral testing, looking for the pathogen in the person and looking for evidence of it. That's most crucial for personal health. This is, we need to know, are you sick? Are you sick with this virus? And do you have a potential to transmit it to your loved ones or your friends, the people you come in contact with on a daily basis, for example? So that was sort of one step of this epidemic. And now we're moving into another phase of the epidemic, which was really starting to look forward. How do we come out of the epidemic? How do we start emerging from the social distancing that we've all been practicing? And now we're seeing that the real power from a public health perspective often lies not so much in looking for the pathogen inside of a person, but looking at the evidence of the pathogen, because that evidence, the evidence that that pathogen did happen and did infect somebody, because the duration over which we can look is so much longer. And this is looking at antibody tests. With an antibody test, we can ask not, are you infected today with the virus, but have you been infected potentially in your lifetime with the virus? And that becomes a very powerful tool, more so for public health than for individual level health. Both of them interplay and interweave in crucial ways in both regards. But if we break it down, we're moving into a public health phase of this epidemic, in part out of necessity. The time for containing and controlling the virus is kind of passed us a long time ago in terms of containing it with sort of individual level testing as our means to really control spread. Now it's more about big public health efforts. And that was, for example, why we all had to social distance, because the virus got out of control. And the only way to contain it is just to have everyone stop moving. But now to emerge from this, testing is going to be a crucial piece. We need to know, where has the virus already hit? What populations are completely susceptible, and potentially what populations is it moving the swiftest through? So for example, New York City, we find from antibody testing that it's maybe hit 20 or 30% of the city, which is an enormous number that we wouldn't necessarily have easily discovered with just antigen or viral testing itself. So both of these have become crucial pieces to how to fight this virus. And we can discuss more about particulars afterwards.
Thank you for that overview, Michael. And as you mentioned, serology and antibody testing, this is where Galit comes in, and you've had to sort of reinvent your life recently. And I hear that you're running one of the largest serology efforts in the state right now at the Broad. Can you tell us a little bit about how that came about and how you're managing this, and what is the output of that effort?
Sure. So I remember in January, I don't know if you remember this, Michael, but we got an email very late at night in like the middle of January from a colleague of ours, Jaap Goudsmit, who used to be the CSO of Crucell before it moved to Johnson & Johnson. And he let us know that he had an antibody that cross-reacted with CoV-2. And so I remember that night very specifically, because I remember that I didn't stop for a second, and I went out and I cloned that antibody. I had it sent out for that gene block, and I made that monoclonal antibody, which is essentially the first ingredient we needed to build an ELISA. So at the time when that happened in January, I think that we hadn't seen the spread really happen out of China, but we knew it was coming. And I guess as a bit of a viral hunter, but like Michael and I kind of like to chase pathogens, we got ready and we were prepared to start doing some deep immunology to understand how what was going to be protective against this infection. What we realized soon after in February, when things started popping in other parts of the world, is that we just needed better tools. There was just no way to track this pathogen. And so what we essentially did is to take these little ingredients. We had a monoclonal antibody that gave us a way to basically build a standard curve. We also had chunks of the sequence, so we could now build antigens from from the virus itself, as well as from related coronaviruses. And we started building assays. And then we realized very quickly, as while everybody else was building up all these RNA tests, there were no antibody tests available yet. And it wasn't really until like the end of February or beginning of March that people started talking about how we needed antibody tests. And so that was really, I think, quite frustrating. And I think Michael and I really got together and started thinking, well, forget all the fancy immunology that we typically do with all these components that we had been building because of this emerging pathogen. But we just went back to meat and potatoes and started building an ELISA, right? It was, there were all these fancy companies out there building, all kinds of interesting devices. And we just said, well, we just need something that is scalable. And more importantly, that was not going to be supply chain dependent based on the troubles that others had experienced, including Michael, with the scaling up of the RNA tests. And so we just built this ELISA. What happened is very quickly, because of that, is we started realizing that we had to redirect all research in the lab because there was just such a demand for testing. And so we did that very quickly. We basically scaled up the ELISA. We worked with Mass General Hospital Pathology to make sure that they could go for an EUA. We, on the research side, built up GCLP qualification so we could essentially do clinically relevant testing. And now we're in the process of validating that assay as well. And now using that, we can do serodiagnostics or serosurveillance, and as well as adapting now to dry blood spot, which Michael has really kind of encouraged us to do so that we could really go at large and really scale up at a population level. So it was really, I never thought I would go back to ELISAs. I always thought we would go forward in technology development, but it really is a necessity that just has become, loud and clear. And so partnership has been key, and that's where we've been moving.
So what types of samples are you processing? Is this now a research study that you're doing, or are you already looking at population levels, or who is sending you samples right now? I presume it's not only your own lab fellows right now.
No, we're not allowed to do those. Actually, there's IRB restrictions, so privacy is always an issue. So what we're getting essentially right now is serosurveillance studies. And so these are all IRB approved studies from all across the country. We're getting samples from where individuals are trying to do serosurveillance in particular populations. There's a number of back to work studies that we are supporting where there are individuals who are still in the workforce and they're trying to understand what is essentially the distribution or what is the level of actual exposure within their population. We're also in the process of working with a number of healthcare worker studies where we're trying to understand at the front line what is the level of exposure and seroconversion for those who are obviously critically important for our efforts in the hospitals. And we've also begun to work with Michael to understand community-based transmission and seroprevalence. So we have a certain capacity of what we can do, but we're already receiving, 5 to 10,000 samples a day. So I mean, the capacity is just really, it was not there before, and it's something that we've had to scale up. So it's pretty intimidating.
Do you still get to do any interesting work at the same time?
I would not. I don't think my fellows and my students would continue if they couldn't do some cool experiments on the side. And so we are still doing some research to try to understand biomarkers of disease progression. And so using our own technologies that look at mechanisms of action of antibodies, we are doing that on the side. But our primary activities right now are exclusively ELISA.
Yeah, I need to correct that, right? Like it's very important work that you're doing.
It's very important.
It's not pushing the boundaries of your scientific
it's pushing the boundaries of policy, and that is the way that we look at it. It's really essential, and it is something that we could not be a part of, and we feel very privileged to be part of that effort. But it really is an interesting redirection of effort.
Right. Thank you. Irene, you run a startup that's venture funded and small and nimble, and we had been talking a while ago about your assay for Zika and dengue. Now I hear that you've also joined the fight against COVID. And so tell us a little bit about how that came about and what that means for a little company like yours.
Oh, sure, and thank you for this meeting. It's great. Just like the panelists have said, our lives probably have changed a lot. So the same thing for E25 Bio and all the virologists in this company. So basically what happened is that around February as well, we were making viral detectors for other viruses. And we also saw that it was imminent that this is going to be the next epidemic, the next pandemic. So we went ahead and tried to see, okay, do we have time to make our own antibodies, which is what we do. We make our own monoclonals to functionalize gold nanoparticles, and make high throughput lateral flow. But of course, there was not going to be enough time. So I remember the day that I went to find out what are the antibodies that are there in the market that detect COVID-2. And literally there was one. There was one antibody. So we did all the contacts. We found about 20 antibodies. We screened them, and we were able to make a pretty decent test to the point that now it's in commercial state. And now we started the clinical trials. So that's how things happened, way too fast for any human being. This is a big challenge. So, and our lives are—
How can you afford to do that? Presumably you have a research plan, a budget, and some workers on this, and now it's something entirely different. So how does that work?
Yeah, so that's when some people who think out of the box pop, and those are venture capital people that call us. And they said, take a plane, come right now and talk to us now. We did. And we got some funding to only do coronavirus testing. And I remember the day I was talking to this investor, and he said to me, guess what? You're going to stop everything you're doing. It's like, how could it be? I mean, yes, you will. I said, really? And it was exactly like that. We had to stop all the Zika or the dengue, the chikungunya, the Ebola, and we already have a product going into market for dengue in Colombia. And we basically stopped everything. And here we are, hopefully going into market with a test that could be useful.
So tell us a little bit about that test, because you and I have been talking about that in the past. I would, as sort of someone running a big organization here, I'd love to have a way to test people when they come in so that we know that there's no virus on site and that we don't have to, go through all of these hoops of distancing.
Totally. So as we just heard very eloquently, it's very important to have memory and see who had the disease and who didn't. And that's very good to have an IgG/IgM test. Nevertheless, you also will always need an antigen test or a PCR that could be massified, but we know that the difficulties of doing that. So what we thought of how our test would work is the following. The test is so nimble and so easy that you could apply it if you want to every day. The test is basically a strip, it's a dipstick, or it has the form of a lateral flow. This is our dengue test. And all you see is a band that appears to your eye, right? And it tells you whether or not you have the disease. And that is possible with coronavirus. The beauty of doing something like this is that the window of optimal detection, it's not just one day, like it happens with PCR, it could be multiple days. So hopefully we hit that wonderful window in which it's about the first 7 days of disease, around there. If you also look at how do we test, we could have a chance to do a nasopharyngeal, which is like, a little bit cumbersome, or you could have a spit, which I believe is 0.5 ml for a PCR to be successful, or you could have a nasal or an oral pharyngeal. So it doesn't matter where the virus is, it includes viremia. So what we found, and this is something that people with more experience could tell us, is that some of our people that we tested, when they're febrile, they express virus in the serum. And this test is also very good on detecting that. For people coming back to work and knowing whether that day they are carrying the virus, even though they don't have fever, then the test will be also very useful. And that's where we thought, okay, we ought to do this. We have to push this as much as we can. And hopefully from clinical studies, we could go to surveillance studies, which is what Dr. Mina and others are doing, in which you basically ask in the population, what are the chances to get a positive test to be positive? So that's how it works.
Thank you, Irene. I hope you'll be producing these tests quickly and cheaply to send me some trial samples first. Michael, back to you. Can you tell us a little bit about how the testing strategy is going to influence policy in Massachusetts? I know that you are part of these bodies that advise our local government on how to best set the state up to emerge out of this. Where do we see testing fall in that? And are we anywhere near the capacity and even the understanding of what it means to then implement that and go back to work safely?
Yeah, so there's certainly two pieces. One of the main discussions is surrounding once we go back to work, can we test people? What are the right strategies for testing individuals to get them back to work to both detect outbreaks at the community level, which is one approach, but then also to more at a very localized level, like in the workplace, in the office, in the laboratory, wherever people might work, to actually detect whether somebody could be a potential vector for transmission on a daily basis. And so these, in many ways, the decision on how to move forward is going to hinge in part, for example, on how successful companies like E25 Bio are in terms of coming out with very rapid, very cheap to produce and use tests for the virus. If there can be, for example, the type of test that exists where people can plan to test themselves every single morning, using a strip that is low cost, because we can't reasonably expect people to spend $50 a day like the PCR test. So it would have to be very low cost. And if we can get there and produce millions of these types of test strips, which I think these companies are working to do, then that can inform one direction where we start to use testing as a true sort of daily part of our existence before going to work. If you're positive, then you don't go to work that day, and maybe you stay at home until you have multiple positive samples in a row. So that's where testing in that sense might come in. There's also going to continue being PCR-based testing from swabs and saliva. And maybe other approaches might be to pool samples together. So everyone swabs themselves each day, for example, leaves the swab in a tube, and then all of those get tested together. For that, that's a different approach that could be very resource efficient and could potentially work. And so those are some of the tests the ways that we're thinking about using testing for the virus. There's a whole different question about using antibody testing to open back up. And this is to identify places where risk is highest. Where should we be placing the most resources? I think even if Irene's company and other companies can get these types of tests to be widely available, they still will be, it still will not be the type of thing that we can assume that every single American will have a nonstop supply of these tests. So we have to think about ways to do this from a public health perspective that is going to be truly sort of long-term solutions. And part of that is recognizing where are the gaps in immunity, what companies or businesses or communities haven't had major outbreaks, or using antibody tests almost as a surrogate for virus. So people's antibodies develop within about a week after they have been infected with the virus. So you can get, from an antibody test, you can not only get recent exposure, but you can also know long-term exposure in the past. So that becomes very powerful if, for example, we want to start monitoring whole communities of people using representative sampling. For example, we might take 300 people from a community who we feel represent the community at large, and we monitor them maybe every week or maybe every month for antibodies. And we can then use those antibodies to detect whether an outbreak is emerging in a community. And then we deploy lots of public health resources and contact tracers and all these very expensive resource intensive efforts to that community and work to prevent the outbreak before it spreads further. So that's sort of at a much more community level how antibody testing is going to be crucial. And really it is one of the more powerful public health tools we have in general, because it can give both history, which gives you some sense of absolute risk to that community in a way, and then it also gives almost up-to-date information on whether the virus is spreading. And the real crucial feature is to follow people longitudinally. What we mean by that is sample the same person over time and sort of enroll people into cohorts. And if we enroll people into cohorts, then we can actually see when somebody moves from negative to positive. It becomes a very powerful public health tool to be able to use to understand how swiftly a virus is moving through communities. Those are the types of things that we wish that we had set up years ago. And a lot of us in the public health space have been saying this for years, that we need to create immunological observatories and things along these lines to be able to just monitor the population. But public health has been largely underfunded for decades. And so these weren't set up. But I think now we're going to see a real change in behavior and where the policies kind of put the funds. And I think we will start having true sort of population representative cohorts being followed, maybe for coronavirus, maybe for all different viruses in the future. Things like West Nile virus and dengue will also be followed in this country, to just kind of constantly monitor these and have the setups in place before outbreaks really begin.
You paint a nice picture of us having the capacity to do all of this. I think Galit sees the daily fight that we don't, right? Like where do you see the capacity come from? Can you just clone what you have and do more of that? Or like how can we get to a point where we can test 10 million people several times per year? Is that even feasible, or is that impossible in the way that we are set up as a non-centralized country? Galit, where do you see this going?
So I don't think it's impossible, Johannes. I think that if there has to be a will. And I think that, as Michael mentioned, I think that there is a need on the public health side, and there is interest now, given what is happening with the, with this particular pandemic, to really begin to scale up and to offer these kinds of resources or this kind of technology to the entire the entire country. And so I think that what's happening is we're starting to realize that this is really a matter of private-public partnerships. And that's the only way this is going to work, right? There is potentially less interest from a commercialization standpoint for building ELISAs or some types of diagnostic tests. They've never been a huge cash cow when it comes to venture investments, but the need and the public health benefit, it outweighs in this kind of crisis what kind of return on investment you get from those kinds of technological developments. So I think that what's happening now is, I mean, it's really a shame that it needed this type of a crisis to make that clear to the world and to the US community and the US market. But I think that is clear. And I can tell you that between Michael, myself, and I'm sure Irene, we're having these discussions, you know, every day. And we're trying to come up with the right solution for the problem so that we can build the capacity and make it so that it's not just twice a year, but it could be weekly, it could be daily, it could be, you know, even, you know, at whenever it's needed. And we just have to have this pandemic preparedness pipeline or infrastructure set up in a way that is accessible to all those who want to access it. And so I think that that is coming. And if you ask us back in maybe a couple of weeks, we'll have an answer for that solution for you.
We root for you for sure. We are getting a bunch of questions from the audience. I'm seeing them coming in on my ticker here. The first one is about how can we introduce these tests to the general populations, and sort of what is the approval path? And maybe, Irene, you can tell us a little bit about your experience trying to get your own test now from sort of prototype into getting it permitted and approved. What are even the words to use here?
It's a great question because FDA, of course, will give you a lot of mandates, very important, but also it requires that they also evolve fast. And I say that because we've been saying that our tests have to be or will have to be in the hands of people. And when you went to talk about regulatory issues on that, they say, well, it has to be in the context of a hospital. It has to be a clinical setting, which limits the whole point of testing a huge population. And therefore, we will see a lot of interactions in the next years, which by contributing between companies and FDA, will come up with ways of putting it in pharmacies or putting it at homes, which is a discussion that hasn't even started. Only HIV, I think, and gonorrhea and others, but H1N1 or influenzas, they're not at home, and then COVID is not at home. So all that is going to evolve. So there are many ways in which I could tell you that the process that we had taught us how to do that. We had to apply for an EUA as an emergency use authorization, which we did. And we found out that there's only 1 or 2 labs in the whole country that could validate our antigen test for 13 different pathogens to show that there is no cross-reactivity, and 7 other pathogens that would not interfere on a simple test, right? And on top of that, there's very few resources to do this. So my comment is that our own experience had taught us that it's doable, that you can put things under FDA approval process. It's painful because you have to generate also collaborations with hospitals and doctors to get the data that FDA asks for. And then more and beyond, you then have to put it in the hands of people, which it's beyond the hospital. So all those will happen. And I think that part of COVID-2 post era is that we probably have a better health in the world because we will all work to be reporting in a very real-time manner and at homes. So that's my take home.
Yeah, I love your optimism. That's a true entrepreneur speaking here. We'll all get better out of this. That's what we like to hope.
We have to.
For all of you, another question from the audience here. Is there any more data on whether or not people develop immunity once they have been infected, and maybe how long is that immunity good for? Michael and Galit, do you have answers for that question? It's one that moves us all.
Well, so there's different ways to look at that question. We know that people develop antibodies, which are by definition part of the immunological response. And we know the kinetics of the virus as it infects somebody and they clear the virus. match what we know about respiratory viruses. So we anticipate that people will have pretty decent immunological responses to this virus that should, in all likelihood, serve to protect them after they have been infected. There's two big unknowns, though. One is, how perfect is that immunity? So is it absolute? For example, when somebody gets measles, we generally don't consider them at risk for measles a second time, potentially through their whole lifetime. But coronaviruses are very different. These are seasonal viruses. We know that people get them more than once. We get even the same strain more than once over a duration of a lifetime. So we're trying to work out, even if somebody has antibodies, does it mean that they're fully protected or partially protected? And if they're partially protected, which might very well be the most likely scenario, could they still potentially transmit the virus afterwards? And that's where we have to just do, with a lot of population level studies are getting set up now, where we are we're taking people potentially who have been infected or at the point of their infection, and then measuring their antibodies, for example, with the assays that Galit has been working so hard to develop, and then following them in time over months, and these studies will actually have to go on for years to recognize, do people who get infected today and have antibodies, is there a level of antibody that actually causes them to have full or partial immunity? And what does that look like in the future? Does it mean they don't get sick but can still spread? Does it mean they do get sick? And is it different for kids versus adults? And does it wane after 6 months or after a year or after 5 years? And these are going to be we're in this for the long haul. This is a new virus that's infecting humans, and we're going to have to be studying it for years to understand these questions.
Yeah, Galit, go ahead.
I could say one thing. So other than thinking about the human data, there are animal studies out there. And so there's at least one study that's published right now on medical archives, which is a small study out of China, where they took rhesus primates, rhesus macaques, and they infected them with the coronavirus. The animals had some disease, and they re-exposed them a short while afterwards. And what was fascinating about the data from that study, and I have to say that there are a number of additional studies that are replicating this currently here at Harvard, but also in other parts of the country and the world, is that the animals do not seem to experience pathology again. So they don't seem to have this deep lung pathology or infection, but they do re-experience viremia in the nose. So I think what this is telling us, this is really quite similar to the reports that are coming out of Korea, where there are people who are testing positive again with their RNA tests after nasopharyngeal swabbing, even though they have antibodies. So they've recovered and then they can be RNA positive again, which led to this kind of fear in the population that maybe reinfection is possible. I think what the data is telling us is that you can be recolonized, but you may not be reinfected with disease again. And so I think it's really important for us to understand what that means. And as Michael said, this is the need to do these long-term studies to understand to what degree do you get recolonized? Is there anything like sterilizing immunity for this virus, which does not seem to be the case for other coronaviruses? And then how do we use the tests if that is the case? So if we know you can be recolonized, but you have antibodies, then the question really is what is an antigen test or an RNA test going to tell us with respect to our antibody measurements? So I think it is a comforting thought, that there is some sort of short-term transient immunity. How long that lasts is to be determined. But then it really starts to make us think a little bit about what the different measurements mean.
There's a question, I think, that goes along those same lines that asks, so if this becomes seasonal, will we have to develop new tests every year? Will mutations affect your testing? Will antibodies be the same for seasonal new variations of this strain? Is there even an understanding of how seasonal would look like in this coronavirus?
Michael, you want to take that one?
Well, so that's where the question of waning immunity comes in, and are the antibodies going to change from year to year? Probably, if the virus doesn't change, the antibodies probably won't change. But we know that that there very well might be, for example, some cross-reactivity between seasonal viruses and this virus. And so we're going to potentially see maybe a huge wave this year. It could even potentially impact or decrease the size of this coming year's seasonal coronavirus wave of infections. But we might end up seeing things that sort of we see as sort of reciprocal events from year to year. We might end up seeing one year you have one big seasonal coronavirus, and then a year or two after you see a different one come up. And so we just don't know, and it will depend in part on if the waning of the antibody response happens over a period of 6 months to a year, then we can anticipate seeing potentially annual infections, but probably in a lot of people, these antibodies might last a longer period of time, and we might end up seeing, especially when you add cross-reactivity into the equation, that it won't just be year on year and year we keep getting beat by this virus, but we'll end up seeing it just become part of the seasonal repertoire.
And I would, this is very important because I would add that because of the forecast that we're seeing coexisting with these viral infections, it's so important to point toward therapeutics. That how come we're so behind? And there's no reason. Scientifically speaking, we could have beautiful therapeutics for sure, but we don't have them because the funding and because the lack of interest. So all that, again, toward the optimism of having the scientific community, medical community to find those new therapeutics, perhaps.
So maybe I can say something about the issue about how the tests are going to do in the next year. So I think that there is some variation in the virus that's popping up. The variation is happening largely, it looks like, in some of the nucleocapsid regions. And so most of the tests currently will most likely be agnostic, since many of the antibody tests are focused on S and on the receptor binding domain of the spike antigen, although there are some nucleocapsid-based ELISAs. But again, the variation is so small, it's unclear it's going to have major consequences on the overall structure of the antigen, but that's to be determined, and that's already underway. I do think it's important to note one thing is I think that, neutralizing antibody tests, right, which we've not discussed at all on this particular call, those are going to be much more sensitive to mutations in the virus that are going to try to get around antibodies that can block infection. And that, was seen in SARS-1 cases back in the early 2000s. And so I think that we are paying attention very closely in the virological societies and groups to look at where these mutations are arising. But the neat thing is, I think with the antigen tests and with the antibody tests, we're really talking about polyclonal antibodies that can bind all over the surface of these molecules, as opposed to one particular footprint that might be essential for blocking infection. So, but we are always thinking about this. And I think that, as the viral sequencing goes on and we see where the amino acid variation is occurring and how that impacts the three-dimensional structure of the virus, we can very rapidly adapt and change those antigens in the assays and increase the resolution.
Is it for the rapid response? I read the New York Times report this morning about Feng Zhang's CRISPR-based Sherlock diagnostics that we know of here. Will they be able to more rapidly adapt to mutating a repertoire of the virus, or is that not relevant?
One of the nice things is of that approach is it could, as long as it's recognized and as long as the mutations are recognized quickly, then certainly that approach could be adapted appropriately in pretty short order. It would still require, you'd actually have to go through, if you start changing the assay to match certain nucleic acids, you'd have to go back to the FDA again and get it reapproved each time you decide to change it. From an FDA regulatory perspective, things have become a little bit looser with the EUA's approach lately, but normally every little piece of an assay that's an FDA approved assay has to be very tightly regulated. And even the smallest change in protocol or type of plastic requires an update to the protocol with the FDA. So it's not something you want to do frequently. So unless it's somehow built into a new model for how the FDA approves assays, if there was like some rolling sort of adaptive response to the virus, but that would require a whole new framework.
And the same applies for PCR. I mean, the PCR that we see, it's based on a probe. And if you have a mutation in the middle of a probe, you're going to compromise the PCR. So it's kind of similar, right?
Yep.
PCR.
We only have a few minutes left, but with the soapbox that we have now for scientists talking to politicians, what is like each of yours message that you would want them to take, and what can we do as scientists to improve our society's response to this challenge and to future challenges? Maybe we'll take a poll from each of you.
I could start by saying that I'm really concerned in which federal funding has been distributed lately. I don't even understand how that decision-making is done. It seems like there's a lot of it going toward molecular testing and whatnot, but definitely a huge gap. And any politician hearing or anybody else different than us, the funds going to hospitals to test things is totally lacking. So if you have companies that are trying to put forth a new technique or a new test, there's no way that the hospital could then take all that into their hands in terms of cost. So one thought is, in my opinion, is can we redirect some funding specifically to stream all the testing, meaning panels, funding to hospitals, etc., so that we have very fast turnaround in testing. That's one thought.
So maybe I can go next, Michael, and you can have the last word. So I think that, I guess my feeling is that, it wasn't a surprise when this outbreak happened, right? It was bound to happen. And in the infectious disease community, we're constantly, battling a new pathogen every so often. It doesn't go away. There's some pathogens like dengue that, are persisting in certain places, but we see them spreading around the world. And so I think my message and, my feeling has always been is that, there's very little attention paid to the development of these diagnostics, and diagnostics have a profound and impactful role in the global economic market of trying to make a difference in health. And so putting a little bit of emphasis on the impact of diagnostics, whether they actually, result in a huge increase in revenue or not, the one thing that is clear is it is an incredibly important increase in revenue on lives saved. And so I think that this is a new dialogue that is happening. And so hopefully we can put the same emphasis on developing that as we do into checkpoint inhibitors and therapeutic vaccines. They're going to have an important impact. And I think that that is something that is, really getting some traction. And so we're really excited to be part of the dialogue.
Thank you.
Yeah, and I think this goes a little bit towards what Irene was saying. So we don't have, in particular, because we don't have centralized healthcare in this country, all of the testing and the way that testing has been set up has been completely centered around individual level health and not around public health efforts. So we don't have any reasonable public health laboratories. We have very specialized public health laboratories that can do a small number of tests at the state and national level, but we don't have a system in place to actually deploy testing at the scale that's really needed for any sort of public health effort. And all of the machines, all of the instrumentation, all of the CLIA labs are generally in hospitals or clinics or our LabCorp and Quest and these types of labs that take samples from patients. And that's a key. We have to think more broadly about, is there a way to not only take samples and test patients, but actually to test the public who are not sick and who are just participating in longitudinal cohort studies and public health programs. And that requires a whole new set of infrastructure and tools and a whole new model of laboratories that can actually run large numbers of tests without doctors' notes, for example, that are part of just big public health programs. And so far we don't have the infrastructure for that in this country. And I think that if we were putting together a comprehensive plan for preparedness in the future, or to respond to this particular virus, it has to include a new model of everything from setting up these facilities to actually paying for the tests that are going to be done. Who pays for them if not Medicare and Medicaid? and private health insurance, this funding has to come from whole different pots of money that generally don't exist. And I think that we'll see them begin to exist more as a result of this virus, but these are the types of real structural changes that have to happen to make preparedness a real thing.
Thank you, Michael, for the summary, and I do hope that we'll learn from this that will have some positive effects on our health care system in general. It is bringing home the need and the demonstration that everybody is vulnerable. I want to thank all of you for making time this afternoon, for sharing with us your experience and wisdom around this. Very interesting comments. Awesome to have access to such experts like you are. Thank you again from all of us here at Lab Central and from the audience. With that, we will cut over to our placeholder picture and invite the audience and you, panelists, if you're interested in joining us next week for our panel on reopening. We'll be having a discussion with business leaders about how they are preparing their own companies and employees to coming back to work after the initial phase of social distancing and what measures they are resorting to. And then the week after, we're going to have a review of sort of the big picture in light of public health questions. Thank you all for joining us this afternoon. Stay safe, stay home, and I hope to see you all again next week. Bye-bye.